Tumor suppressor gene. Published by Wikipedia. A tumor suppressor gene, or anti-oncogene, is a gene that regulates a cell during cell division and replication. If a cell grows uncontrollably, it will result in cancer. When a tumor suppressor gene is mutated, it results in a loss or reduction in its function. In combination with other genetic mutations, this could allow the cell to grow abnormally. The loss of function for these genes may be even more significant in the development of human cancers, compared to the activation of oncogenes. TSGs can be grouped into the following categories: caretaker genes, gatekeeper genes, and more recently landscaper genes. Caretaker genes ensure stability of the genome via DNA repair and subsequently, when mutated, allow mutations to accumulate. Meanwhile, gatekeeper genes directly regulate cell growth by either inhibiting cell cycle progression or inducing apoptosis. Lastly, landscaper genes regulate growth by contributing to the surrounding environment, and when mutated, can cause an environment that promotes unregulated proliferation. The classification schemes are evolving as medical advances are being made from fields including molecular biology, genetics, and epigenetics. History. The discovery of oncogenes and their ability to deregulate cellular processes related to cell proliferation and development appeared first in the literature as opposed to the idea of tumor suppressor genes. However, the idea of genetic mutation leading to increased tumor growth gave way to another possible genetic idea of genes playing a role in decreasing cellular growth and development of cells. This idea was not solidified until experiments by Henry Harris were conducted with somatic cell hybridization in 1969. Within Harris experiments, tumor cells were fused with normal somatic cells to make hybrid cells. Each cell had chromosomes from both parents, and upon growth, a majority of these hybrid cells did not have the capability of developing tumors within animals. The suppression of tumorigenicity in these hybrid cells prompted researchers to hypothesize that genes within the normal somatic cell had inhibitory actions to stop tumor growth. This initial hypothesis eventually led to the discovery of the first classic tumor suppressor gene by Alfred Knudsen, known as the RB gene, which codes for the retinoblastoma tumor suppressor protein. Alfred Knudsen, a pediatrician and cancer geneticist, proposed that in order to develop retinoblastoma, two allelic mutations are required to lose functional copies of both the RB genes to lead to tumorigenicity. Knudsen observed that retinoblastoma often developed early in life for younger patients in both eyes, while in some rarer cases retinoblastoma would develop later in life and only be unilateral. This unique development pattern allowed Knudsen and several other scientific groups in 1971 to correctly hypothesize that the early development of retinoblastoma was caused by inheritance of one loss-of-function mutation to an RB germline gene followed by a later de novo mutation on its functional RB gene allele. The more sporadic occurrence of unilateral development of retinoblastoma was hypothesized to develop much later in life due to two de novo mutations that were needed to fully lose tumor suppressor properties. This finding formed the basis of the two-hit hypothesis. In order to verify that the loss of function of tumor suppressor genes causes increased tumorigenicity, interstitial deletion experiments on chromosome 13q14 were conducted to observe the effect of deleting the loci for the RB gene. This deletion caused increased tumor growth in retinoblastoma, suggesting that loss or inactivation of a tumor suppressor gene can increase tumorigenicity. Two-hit hypothesis. Unlike oncogenes, tumor suppressor genes generally follow the two-hit hypothesis, which states both alleles that code for a particular protein must be affected before an effect is manifested. If only one allele for the gene is damaged, the other can still produce enough of the correct protein to retain the appropriate function. In other words, mutant tumor suppressor alleles are usually recessive, whereas mutant oncogene alleles are typically dominant. Proposed by A.G. Knudsen for cases of retinoblastoma, he observed that 40% of U.S. cases were caused by a mutation in the germline. However, affected parents could have children without the disease, but the unaffected children became parents of children with retinoblastoma. This indicates that one could inherit a mutated germline but not display the disease. Knudsen observed that the age of onset of retinoblastoma followed second-order kinetics, implying that two independent genetic events were necessary. He recognized that this was consistent with a recessive mutation involving a single gene, but requiring biallelic mutation. Hereditary cases involve an inherited mutation and a single mutation in the normal allele. Non-hereditary retinoblastoma involves 2 mutations, 1 on each allele. Knudsen also noted that hereditary cases often develop bilateral tumors and would develop them earlier in life, compared to non-hereditary cases where individuals were only affected by a single tumor. There are exceptions to the 2-hit rule for tumor suppressors, such as certain mutations in the p53 gene product. p53 mutations can function as a dominant negative, meaning that a mutated p53 protein can prevent the function of the natural protein produced from the non-mutated allele. Other tumor suppressor genes that do not follow the 2-hit rule are those that exhibit haploinsufficiency, including PTCH in medulloblastoma and NF1 in neurofibroma. Another example is p27, a cell cycle inhibitor, that when one allele is mutated causes increased carcinogen susceptibility. Functions. The proteins encoded by most tumor suppressor genes inhibit cell proliferation or survival. Inactivation of tumor suppressor genes therefore leads to tumor development by eliminating negative regulatory proteins. In most cases, tumor suppressor proteins inhibit the same cell regulatory pathways that are stimulated by the products of oncogenes. While tumor suppressor genes have the same main function, they have various mechanisms of action that their transcribed products perform, which include the following. Intracellular proteins that control gene expression of a specific stage of the cell cycle. If these genes are not expressed, the cell cycle does not continue, effectively inhibiting cell division. Receptors are signal transducers for secreted hormones or developmental signals that inhibit cell proliferation. Checkpoint control proteins that trigger cell cycle arrest in response to DNA damage or chromosomal defects. Proteins that induce apoptosis. If damage cannot be repaired, the cell initiates programmed cell death to remove the threat it poses to the organism as a whole. Cell adhesion. Some proteins involved in cell adhesion prevent tumor cells from dispersing, block loss of contact inhibition, and inhibit metastasis. These proteins are known as metastasis suppressors. Proteins involved in repairing mistakes in DNA. Caretaker genes encode proteins that function in repairing mutations in the genome, preventing cells from replicating with mutations. Furthermore, increased mutation rate from decreased DNA repair leads to increased inactivation of other tumor suppressors and activation of oncogenes. Certain genes can also act as tumor suppressors and oncogenes. Dubbed proto-oncogenes with tumor suppressor function, these genes act as double agents that both positively and negatively regulate transcription. Epigenetic influences. Expression of genes, including tumor suppressors, can be altered through biochemical alterations known as DNA methylation. Methylation is an example of epigenetic modifications, which commonly regulate expression in mammalian genes. The addition of a methyl group to either histone tails or directly on DNA causes the nucleosome to pack tightly together, restricting the transcription of any genes in this region. This process not only has the capabilities to inhibit gene expression, it can also increase the chance of mutations. Stephen Balin observed that if promoter regions experience a phenomenon known as hypermethylation, it could result in later transcriptional errors, tumor suppressor gene silencing, protein misfolding, and eventually cancer growth. Balin et al. Found methylation inhibitors known as azacitidine and decitabine. These compounds can actually help prevent cancer growth by inducing re-expression of previously silenced genes, arresting the cell cycle of the tumor cell and forcing it into apoptosis. There are further clinical trials under current investigation regarding treatments for hypermethylation as well as alternate tumor suppression therapies that include prevention of tissue hyperplasia, tumor development, or metastatic spread of tumors. The team working with Wadjet have investigated neoplastic tissue methylation in order to one day identify early treatment options for gene modification that can silence the tumor suppressor gene. In addition to DNA methylation, other epigenetic modifications like histone deacetylation or chromatin-binding proteins can prevent DNA polymerase from effectively transcribing desired sequences, such as ones containing tumor suppressor genes. Clinical significance. Gene therapy is used to reinstate the function of a mutated or deleted gene type. When tumor suppressor genes are altered in a way that results in less or no expression, several severe problems can arise for the host. This is why tumor suppressor genes have commonly been studied and used for gene therapy. The two main approaches used currently to introduce genetic material into cells are viral and non-viral delivery methods. Viral methods. The viral method of transferring genetic material harnesses the power of viruses. By using viruses that are durable to genetic material alterations, viral methods of gene therapy for tumor suppressor genes have shown to be successful. In this method, vectors from viruses are used. The two most commonly used vectors are adenoviral vectors and adeno-associated vectors. In vitro genetic manipulation of these types of vectors is easy and in vivo application is relatively safe compared to other vectors. Before the vectors are inserted into the tumors of the host, they are prepared by having the parts of their genome that control replication either mutated or deleted. This makes them safer for insertion. Then, the desired genetic material is inserted and ligated to the vector. In the case with tumor suppressor genes, genetic material which encodes p53 has been used successfully, which after application, has shown reduction in tumor growth or proliferation. Non-viral methods. The non-viral method of transferring genetic material is used less often than the viral method. However, the non-viral method is a more cost-effective, safer, available method of gene delivery, not to mention that non-viral methods have shown to induce fewer host immune responses and possess no restrictions on size or length of the transferable genetic material. Non-viral gene therapy uses either chemical or physical methods to introduce genetic material to the desired cells. The chemical methods are used primarily for tumor suppressor gene introduction and are divided into two categories, which are naked plasmid or liposome-coated plasmids. The naked plasmid strategy has garnered interest because of its easy-to-use methods. Direct injection into the muscles allows for the plasmid to be taken up into the cell of possible tumors, where the genetic material of the plasmid can be incorporated into the genetic material of the tumor cells and revert any previous damage done to tumor suppressor genes. The liposome-coated plasmid method has recently also been of interest since they produce relatively low host immune response and are efficient with cellular targeting. The positively charged capsule in which the genetic material is packaged helps with electrostatic attraction to the negatively charged membranes of the cells as well as the negatively charged DNA of the tumor cells. In this way, non-viral methods of gene therapy are highly effective in restoring tumor suppressor gene function to tumor cells that have either partially or entirely lost this function. Limitations. The viral and non-viral gene therapies mentioned above are commonly used, but each has some limitations which must be considered. The most important limitation these methods have is the efficacy at which the adenoviral and adeno-associated vectors, naked plasmids, or liposome-coated plasmids are taken in by the host's tumor cells. If proper uptake by the host's tumor cells is not achieved, reinsertion introduces problems such as the host's immune system recognizing these vectors or plasmids and destroying them, which impairs the overall effectiveness of the gene therapy treatment further. Examples. Retinoblastoma protein. PRB was the first tumor suppressor protein discovered in human retinoblastoma. However, recent evidence has also implicated PRB as a tumor survival factor. RB1 gene is a gatekeeper gene that blocks cell proliferation, regulates cell division and cell death. Specifically, PRB prevents the cell cycle progression from G1 phase into the S phase by binding to E2F and repressing the necessary gene transcription. This prevents the cell from replicating its DNA if there is damage. P53. TP53, a caretaker gene, encodes the protein P53, which is nicknamed the guardian of the genome. P53 has many different functions in the cell, including DNA repair, inducing apoptosis, transcription, and regulating the cell cycle. Mutated P53 is involved in many human cancers. Of the 6.5 million cancer diagnoses each year, about 37% are connected to P53 mutations. This makes it a popular target for new cancer therapies. Homozygous loss of P53 is found in 65% of colon cancers, 30 to 50% of breast cancers, and 50% of lung cancers. Mutated P53 is also involved in the pathophysiology of leukemias, lymphomas, sarcomas, and neurogenic tumors. Abnormalities of the P53 gene can be inherited in Li-Fraumeni syndrome, which increases the risk of developing various types of cancers. BCL2. BCL2 is a family of proteins that are involved in either inducing or inhibiting apoptosis. The main function is involved in maintaining the composition of the mitochondria membrane and preventing cytochrome C release into the cytosol. When cytochrome C is released from the mitochondria, it starts a signaling cascade to begin apoptosis. SWI-SNF. SWI-SNF is a chromatin remodeling complex, which is lost in about 20% of tumors. The complex consists of 10 to 15 subunits encoded by 20 different genes. Mutations in the individual complexes can lead to misfolding, which compromises the ability of the complex to work together as a whole. SWI-SNF has the ability to move nucleosomes, which condenses DNA, allowing for transcription or block transcription from occurring for certain genes. Mutating this ability could cause genes to be turned on or off at the wrong times. As the cost of DNA sequencing continues to diminish, more cancers can be sequenced. This allows for the discovery of novel tumor suppressors and can give insight on how to treat and cure different cancers in the future. Other examples of tumor suppressors include PVHL, APC, CD95, ST5, YPEL3, ST7, and ST14, P16, BRCA2.